Hello and a warm welcome to our webinar today. Really appreciate you joining us for today's topic of real-time monitoring of barrier integrity with TEER and organ on chip. Also, we'd like to thank everyone who submitted their questions in advance. We really used them to shape the content of this webinar, and they will be the first ones that we're going to answer once Sophie's going to be finished with her presentation. Now on to just a bit of housekeeping. So you probably have seen your control panel. There's a chat and a Q&A function. And we would really appreciate if you use the chat function for any queries that you have throughout the webinar, and I will try to get back to them very quickly. And then use the Q&A function for the questions that you have to our speakers that we will attend to after the presentation is finished. So now, without further ado, I'd like to introduce our speakers for today. So first up, we have Sophie Besser. She is part of our R&D team. She's a scientist here, and she holds a Bachelor of Engineering in Biotechnology and a Master of Science in Pharmaceutical Biotechnology. In April 22, Sophie began her master's thesis here on the emulation of an inflammatory bowel disease model, and then she transitioned into a full-time position in August 23. Also joining us today is our CEO and co-founder, Martin. He'll be joining for the Q&A session later on. He's been working and researching with on-chip cell culture applications since 2010, so a very long time. And at Jena University Hospital, he and his co-founder, Knut Rennert, first-hand experienced the need for physiological human organ models that really properly represent the situation in vivo. And since 2021, he's also board member of the European Network for Organ on Chip EUROX. So now with this, I'd like to hand over to Sophie.
Hello, everyone. So first maybe a few words about our company. We provide organ on chip technology and also education around it, training, and we are also a service provider. Our company combines a lot of scientific expertise and comprehensive biology in various fields with the ease of use system, and we also provide lots of community support around it, if you're interested. We are located in central Germany, in Jena, and here you can see a nice picture of our team who's doing all the great work. In this brief webinar, I will first introduce the concept of organ on chip, and then I mainly focus on today's topic, the TEER measurement. We will cover what TEER is and how it is integrated into our organ on chip system using the intestine on chip model as an example. And at the end, we are looking at useful applications of TEER in organ on chip platforms. So, for anyone who's not yet familiar with this technology, what exactly is organ on chip? Here you see a general definition from the European Organ on Chip Society. It describes a fit-for-purpose microfluidic-based device with different chambers and channels, allowing the setup of living organ substructures. This also enables the creation of a controlled microenvironment within the chip. And once the tissue has developed, it can be connected to a perfusion system to mimic then blood flow and nutrient supply, similar to the conditions in the human body. And with this comes also biomechanical stimulation, which then affects receptor activation and downstream analysis or downstream signaling within the tissue, and moves it towards a more physiological and in vivo-like state. And with this, we want to improve the translation of data between in vitro and in vivo and contribute to close this gap. Yeah. So what are the current possible readouts with organ on chip? In this case, especially with our D42 biochip, I can tell you that the D42 platform is highly flexible and you can perform most downstream analysis you know already from standard cell culture. So first it is suitable to sample supernatant at any time point to check for biomarkers such as clinical parameters, or cytokine profiling is possible, or you can check for compound turnover, or viability or cytotoxicity. You can also easily recover the tissue from the chip, either as a single cell suspension, or you can lyse the tissue inside the chip for downstream analysis. But you can also take out the whole tissue for immunofluorescence staining or histological staining. Our platform is designed in the microscopic slide format, so that it easily fits into existing lab workflows, and you can do beautiful live cell imaging. And live cell imaging is also suitable to investigate other parameters like reactive oxygen species formation or mitochondrial activity. And last but not least, you can check for enzyme activity, protein and transporter functionality. There are plenty of kits on the market that are available, and all protocols, or the most of the protocols, are all adaptable to our platform. And you can also use them to do your readouts. But there's even more to get out of your experiment. We can also now monitor real-time tissue barriers. The fundamental aspect of organ-on-chip technology is the implementation of compartmentalized tissue barriers under dynamic conditions, and by recreating these barriers in a controlled environment, the organ-on-chip platforms allow us to better understand their roles and the interactions in the body. But what exactly are tissue barriers, and why are they so important? So tissue barriers are specialized cellular layers like epithelial or endothelial layers that control what can pass between two compartments in the body. In this way, they act as a selective filter. They allow essential substances like nutrients, oxygen, and hormones to pass through. But they also block harmful agents. And in doing so, they help maintain homeostasis and ensuring a stable and a balanced internal environment. Another point is that they prevent toxins and pathogens from reaching and damaging the vital organs or some vital organs, what makes them a key part of the body's defense system. So they're very important, and the key structures of these barriers are tight junctions, adherence junctions, and desmosomes. There are several vascular and also epithelial barriers in the body that are of high importance. For instance, a very well-known barrier is the blood-brain barrier. The blood-brain barrier restricts a molecule transport into the brain, but we also have a gut epithelial barrier that helps us to separate the luminal located microbes in the body to make us not sick, but stay in the intestine and have a beneficial effect on our health. And these are just a few examples. There are many more, but these barriers can be monitored in an organ-on-chip via TEER. And this is what we implemented in our chip platform. So short TEER is the Trans-Epithelial and Trans-Endothelial Electrical Resistance. It is a widely used non-invasive method to assess how well epithelial or endothelial cells form a functional barrier in vitro. And basically the electrical resistance, which is measured across the cell layer, gives us an idea of how intact or differentiated the monolayer is, especially if the cells are forming strong tight junctions. And it's also a simple and fast readout. It's commonly used in static culture, and it gives a first indication of barrier quality before moving on to more complex analysis. Before TEER became more widely used, permeability assays were the standard method to evaluate barrier integrity in vitro. And while there are still valuable tools, comparing both methods reveal key advantages of TEER measurement. So let's take a closer look at how TEER and permeability assays differ, in this case in a transval. So permeability assays rely on a labeled tracer molecule to see how much of this tracer molecule passes really through the cell layer over time. But TEER, on the other hand, uses an electrical current to measure the resistance across the monolayer, which then reflects how tight the cell junctions are. And in terms of timing, permeability assays can take anywhere from 1 to 24 hours, depending on the setup, whereas TEER measurements are much quicker, often just a few minutes or seconds. And when it comes to compatibility, the permeability assays could interfere with cells or drugs, depending on the tracer used. And TEER is a less invasive method. In that sense, it has a limited interference with cells and drugs. But its accuracy can be quite sensitive to experimental conditions. Things like electrode type, as you can see here, the chopstick electrodes, the membrane area, or media temperature, that can all influence the outcome. So both methods have their strengths and limitations, and they are often used together to get a fuller picture of barrier function. But compared to these in vitro methods, like the use of chopstick type electrodes in transfer cultures, the TEER electrodes in our chip platform are incorporated at a fixed position without being movable, and therefore it provides a more accurate and stable measurement with less sensitivity distribution. So that's a big advantage in comparison to the chopstick TEER measurement. And here you can see a schematic of the chip where we equipped our biochip with semi-transparent planar gold electrodes. And in this explosion view, you can see that the electrodes are positioned at the inside of the chip, of course attached to the top and the bottom bonding foil. This enables you to get a readout throughout the tissue and check if the barrier is intact or not. And we also added an additional circuit board that is attached to the chip. And this circuit board enables the contacting of the electrodes and the signal transmission. And here you can see how it looks in real life, how our chip looks in real life from the front, from the top view and the bottom view. That's not all. Also here we laid a focus on ease of use and designed a measurement device for contactless readout. The measurement device can operate up to 3 chips in parallel. On each chip we have 2 models, so we can measure 6 models with 1 device. And the measurement interval can be adjusted from very short intervals in range of seconds up to 1 hour. And here on the left side, you see an exemplary setup, how it can look like in the incubator. And here on the right side is a real-life impression from an incubator setup. Yeah. The signals from the TEER chip are transmitted via Bluetooth to a computer with a data plotter software, and this is of course designed as an open platform, so every open source data plotter software can be used for this. And as TEER is a temperature sensitive technique, we thus avoid biasing the experiment by keeping the incubator closed at all time. And before I present you one TEER use case on our gut-on-chip, I briefly want to introduce our intestinal model and explain it to you. When you see inside the chip, it begins with the top bonding foil on the chip and it ends with the bottom bonding foil, and everything between is inside the chip. There's a porous membrane that compartmentalizes the chamber into a top and a bottom channel. In the top channel, we set up a vasculature, and in the bottom channel, we set up the intestinal compartment. And when we start perfusing it, it also starts to outgrow beautifully in a villus-like structure as we find it in the human body. And in the intestine, there are many parameters that are of interest, but for instance, one is the barrier. And that's why we chose the gut-on-chip for our first experiments with the TEER chip. So how is our model exactly assembled? First, endothelial cells, the UVECs, are seeded into the upper chamber of the chip. And after 48 hours, macrophages are added, and 24 hours later, then the epithelial cells, the CACO2 cells, are seeded into the lower epithelial chamber. And once the entire model has been statically cultured for another 24 hours, both the vascular and the epithelial chambers are connected to the perfusion, which induces the formation of the villus 3D structure. And here in the image on the right, you can see how it looks at the end inside the chip with the planar electrodes. During this model assembly, I mentioned, we also recorded TEER values to see how the barrier in these chips changes every time we seed in new cells. Upon seeding UVECs, the TEER values slightly increased. Seeding then the MVMs led to a small elevation in TEER, but during seeding and formation of the confluent endothelial cilia, the TEER values remained mainly low. A major increase in TEER was then observed after seeding in the CACO2 cells in the bottom channel. They formed a really tight barrier over 24 hours, as you can see at the TEER values. And when we connected the chips to the perfusion, the TEER values at first increased, but then remained stable over 24 hours perfusion, but also at a high point. And further, and very important, we can show that the cell viability is not affected, so it's the same as it is in chips that do not have the TEER electrodes integrated. You can see it here by ATP measurements of the tissue that shows us the biocompatibility of the electrodes. After our standard model assembly, as I previously shown you, the models were perfused for an additional 5 days. Then as shown here, the experimental barrier disruption was performed using EDTA, followed by a recovery phase, which was also monitored via TEER measurements. And the EDTA was applied to the intestinal bottom chamber for 30 minutes. And afterwards, the EDTA containing medium was replaced with fresh medium without EDTA, which then induces tissue recovery within 24 hours. But First, shortly, what exactly is EDTA? How does it affect the cellular barrier? EDTA is a strong metal-ion chelator. It is known to bind magnesium and calcium and thereby disrupting the epithelial barrier. So, as you also can see here, when we treated the model, we saw immediate effects upon EDTA administration, that the TEER values dropped down close to 0, as you can see on the left graph. And when we remove the EDTA, we can then also nicely see that the tissue barrier recovers over the time to the control TEER values. So the TEER measurement, we're able to detect the EDTA-induced barrier induction and also the tissue recovery. We also compared it to a standard technique, as mentioned, for barrier measurement and permeability assay. In this case, the FITC-Dextran. In this FITC-Dextran, we also tested every condition, the control model, the EDTA treated model, and also after recovery time overnight. And we saw that this was not that sensitive to really detect significant changes or small changes in the tissue barrier, as we saw it really in the TEER measurements. So here you can see really the advantage of the TEER measurement in comparison to standard techniques like permeability assay. We can also beautifully capture the EDTA effect via bright field imaging. The light microscope images revealed also cell rounding and detachment from the membrane after 30 minutes of EDTA treatment. And after recovery, you see again the 3D morphology with the villus and the crypt-like structures, as you can see it also in the control model. And these images also show that the electrodes are semi-transparent, so you still have the possibility on a bright field microscope to look at the tissue and to investigate it. As you can see here in the lower pictures, the arrows show the borders of the TEER electrodes under the microscope, so it's really semi-transparent. Of course, to complement this data, we stained for tissue barrier related markers such as E-cadherin or ZO1 in the intestinal cells. Both junction proteins were disrupted after the treatment with EDTA for 30 minutes, which can be clearly seen in the highlighted boxes with the right arrows. And the good distribution of the DAPI signal shows us also that the treatment is affecting the junctional complexes, but not the nuclei integrity. And after 24 hours of recovery, you see similar marker expression as at the beginning, as in the untreated models, the controls. The expression of cell markers in the vascular cell layer showed similar results. We stained for CD31 and phenylreprint factor, and both markers were also disrupted after EDTA treatment. But they could also be restored after a 24-hour recovery phase with the help of a fresh medium. So, to sum this up, we demonstrated here the integration of semi-transparent gold electrodes, and the operation works via simple plug-and-play workflow with the flexible intervals for measurement in our proof of concept study that we run for 2 weeks. We could see the changes that appear when we administer treatment, in this case, for instance, with EDTA. The large electrode area favors also the quantification and the denormalization of TEER values, which is really nice to make it more comparable to other systems or applications, for instance, also for trans health. And the system shows a high biological reproducibility during several experiments we did. We saw only minor changes in TEER levels that we recorded. Everything was pretty close together. So our model offers several key advantages. First of all, our system is completely PDMS-free, which helps to avoid the typical problems with molecule absorption that are often seen in PDMS-based systems. It's designed for simple fabrication, making it easier to manufacture and scale. And the scalable electrode design ensures flexibility for different applications. And with the wireless contact-free readout via Bluetooth, measurements are non-invasive and keeping your sample intact. And the system is also fully compatible with incubator environments and offers intuitive operation that makes it user-friendly for everyday use. And additionally, it is also very easy to sterilize and maintain. Yeah. This slide summarizes the main application of TEER measurements in organ-on-chip platforms in general. First, TEER is widely used to assess the tightness of cell junctions, the permeability of tissue barriers, and the overall structural integrity or the epithelial or endothelial layers of the epithelial or endothelial layers. Second, it can provide a clear indication of how drugs and compounds affect the barrier, including changes in transport mechanisms or how cells respond to pharmacological stimuli. And finally, TEER enables also real-time monitoring of barrier function under disease-like conditions, including here inflammatory responses, infection models, or tissue damage. And this makes TEER a powerful non-invasive tool to study dynamic physiological and pathological processes in a highly controlled environment. So, if you want to learn more, feel free to visit our website, follow us on LinkedIn or Blue Sky. Our general technology is available through the dynamic organ product line, and we also offer both on-site and online trainings, as well as detailed application notes to help you get started or deepen your understanding. And with that, I give to you back, Juliane.
Yeah, thank you for the slides. Definitely a lot of stuff that was also new to me, so very interesting. Now we're moving over to our Q&A session. So I can already see a lot of questions coming in through the Q&A function. But as mentioned, we're gonna first answer all the questions that have been submitted before we started this webinar. So Sophie prepared some slides for this, I believe.
So one question was, is it possible to harvest the cells from the organ-on-chip device? And as you saw in my presentation, yes, it is possible. As you also can see here in the images, even with the electrodes on the bonding foil, the membrane can be removed using a scalpel, or you can recover the tissue from the chip either as a single cell suspension or lyse the tissue inside the chip. And from there, the cells can be processed as needed, for example, for immune fluorescence staining or other things. The second question was, this method in OOC still is unstable and varies. How to achieve stability in TEER measurement? I would say we achieve good stability in our TEER measurements in two main ways. First, we use fixed planar electrodes which stay in place and avoid variation that can happen if the electrodes are moved or not positioned exactly the same each time. And this helps make the reading more reliable. And second, TEER is very sensitive to temperature changes. That's why our system is designed to work directly inside the incubator, and both the chip and the measurement device stay in the controlled environment. And then, thanks to the wireless contact-free readout, we can measure from outside without opening the incubator or disturbing the cells. And as you can see in our results here, the experiment was done 3 times, and the measurements are very consistent, showing that our system provides stable and repeatable data. Another question was if there are examples for both human and animal cell usage. I only can say so far we only tested it with human cells, so no animal cells used before. Then there was a question, I use D42BC002 biochips. Is there a way of measuring the TEER in these biochips? Unfortunately, I have to say it's not possible with the standard BC002 biochip. It requires the TEER upgraded version with the electrodes in it. But since it's still based on the BC002, your existing protocols and workflows do not need to be changed. Another question was, how much is the TEER chip? Do you need any additional material, for example, a voltmeter and cables? And since we currently only offer the chips as a part of fee-for-service projects, I can't tell you the pricing details yet, but we would let you know as soon as they become commercially available, which will likely be hopefully at the end of the year. And in the meantime, we're also looking for beta testers. So if you're interested, feel free to reach out to us. Yeah. And to answer the second question, you don't need any voltmeter or any additional cables. All you need is the chip, the measurement device, and any laptop running an open source plotting software. It doesn't matter which software you use. So you can choose every software for this. What is the influence of holes in cell tissue layer on obtained signal and overall current distribution? I have to say, big holes or gaps in the cell layer can strongly affect the TEER measurements. The current flows through these gaps, and then there's no resistance, which leads to lower TEER values. So you're not really longer measuring the true barrier function. That's why we always make sure the cell layer is fully confluent before we starting the measurements or treatment or manipulating it. Can we develop a simulation of the model to develop the chip response in silico? We haven't worked on that directly yet, but we've had experience with similar approaches. Earlier this year, we published something on this topic in collaboration with Bayer. Maybe you can have a look at this. The QR code will take you to the paper. Yeah, that were all questions who were submitted up front.
Hey, thank you, Sophie. So we're going to start with the question that have been submitted now. The first one that actually came in during your presentation was, why did TEER increase during the perfusion?
It's only increasing because it have changed. We put on the perfusion and something changed inside the chip, but then you saw that it gets stable again, and then there were no other changes. They were really stable.
Okay, thank you. Martin, did you want to add anything?
Yes. It is most likely also a reaction of the cells to the perfusion, to the shear forces, so that the tissue and the cell layer is tightening, and that's why we see an increase in TEER upon perfusion.
Excellent. So the next question is, how many cells can you recover from the chip for downstream molecular assays? If the cell number is not high enough, it may be difficult to perform qPCR or flow cytometry.
This is a bit, thank you for the question. This is a bit depending on the model used. In the intestinal model, we have plenty of cells because of the intestinal layer that is outgrowing. So here we're talking about 6 million cells or more in a total. But for other cells like lung on chip or liver on chip, it's less, but still between 700K and 1 million cells, because the overall culture area in our standard biochip is similar to 24 well, to the size of a 24 well. And so we can introduce quite a lot of material into the chip, and therefore ensuring compatibility with a lot of readouts, not just for cell lysis to get proteins out of it, but also for the biomarker secretion of cells.
Excellent. And the next one is for confocal imaging. Would you provide the protocols for the experimentation?
Of course. Just please get in contact with us, and we can set up a separate meeting and talk about your specific application and how you can realize this. So we're very open to share our experiences here and to guide you through this.
Thank you, Martin. Next one is very nice work. Thank you. I'm new to this area of research. Can real-time TEER be measured in transfer systems, or is it only possible in this type of systems?
So it's also possible in transfer systems, but we are not doing it here. So in our company, we're only doing it in the organ-on-chip system, but a lot of research groups do it also in the transfer.
Thank you, Sophie. Will the probe at the bottom side block the view of the microscope?
I guess my probe, also the second electrode, it's referring to the second electrode of the on the bottom foil of the chip. And no, also our pictures are made with a bright field microscope where the light comes from beneath the chip, so it goes completely through the chip and nothing is interfering here.
Excellent, that's very good news. Next question is from Rita. Hi, I'm new to this topic. How do you sterilize the TEER electrode? 70% ethanol or other approaches?
We sterilize with 70% ethanol.
Cool, short and sweet, I like it. So next one is from Chase. Is the TEER measurement device tuned to a specific frequency based on the electrode material for measurement, or can this be changed?
At the moment, it is tuned to a specific frequency, but I think it can be changed if needed. But yeah, at the moment, it's only a specific frequency.
Cool. So for now, we've actually reached our very last question. So if you've got any more questions, feel free to submit them now. Otherwise, I will likely conclude the webinar after this. So the last question is from Arnita. Is there any clear, trustable source where to look for normalized TEER values for particular cell models. As seen in literature, the difference is too significant due to different TEER setups. So Martin, did you want to take this one, or you're unsure?
To be honest, I'm a bit unsure about a good literature source for normalized TEER values. So we've noticed this also, that this is varying throughout various publications. That's why we put special attention to providing something where you can normalize the TEER values to in reference to the size of the electrodes. But yeah, that's a good question, but I don't have an answer for this. Sorry.
That's all right. We can't always answer everything, but actually, if you take a look in the chat, so Martin wrote something, but also Tim, who was, besides Sophie, the other lead scientist who developed this platform, he's posted a link in there as well. So I believe this will be quite helpful as well. Actually, we've got another question coming in. So from Carlos, do you see alternating current in your TEER setup? If so, what's the effect of different frequencies? Getting very technical now. I see we're getting deeper into the TEER topic here.
So we have someone who's doing this technical stuff, and he was also the guy for the frequency setups and so on. So I can't really answer this question. That was his job.
Okay, that's fine. But we can try and forward this question to him and then just get back to you afterwards. So now that I see there are no questions left and we're well within time, I will conclude this webinar. I thank you a lot for joining us today. We really appreciate this. There was really a lot of interest in this, and I will send the recording after this so that you could share with your colleagues or just revisit some of the slides. And should you have any more questions, you can always reach out to us and we will get back to you. Thank you and goodbye.
Goodbye.